Assalamu alaikum wa rahmatullahi ta'ala wa barakatuh. In the series of lecture concerning the course Applied Pharmacognosy, which is designed for fifth-year pharmacy student, in this eighth lecture, we will talk about the biosynthesis of secondary metabolites. Biosynthesis of secondary metabolites is considered as a chemical process which involve a series of biochemical conversion that result in converting primary metabolite into complex molecule with a specialized function. These complex molecules, known as secondary metabolite, and play a crucial role in plant defense. Also, they have ecological interaction role for plant, and for human they have medicinal application. The process of biosynthesis is regulated by enzymes. They have important role, and there are several factors and reagents that involved in this process. In order to understand the process of biosynthesis for secondary metabolite, this lecture will talk about several subtitles or several topics. Firstly, we will talk about the definition of secondary metabolite, their production and storage. Secondly, we describe the plant cell component that involved in biosynthesis of secondary metabolites. Also, the role of enzymes in biosynthesis. Fourth, we will talk about the basic metabolic pathways that are essential for production of primary and secondary metabolites. Also, we will talk about the relation between secondary metabolite and these basic metabolic pathways. Lastly, in details, we will talk about the origin of secondary metabolite and the basic or the pathways that involved in production of secondary metabolite, their precursors, and important biotic action involved in these processes. One of the important questions concerning production of secondary metabolite, we have to answer the question about the identity of plant secondary metabolites. Or in other words, what are secondary metabolites? Secondary metabolites are organic compounds biosynthesized in the plant to aid their survival. Actually, they are not directly involved in normal growth or development or reproduction of the plant or an organism. In fact, they have ecological function for plant. They're responsible for other plant, pungency, color. Also, they have medicinal application, culinary application, and they have poisonous value. Example of secondary metabolite include the compound glycosides and aromatic compounds such as flavonoids, anthracenoids, tannins, also isoprenoids, and so on. Production of secondary metabolites is a biochemical process which is regulated by enzyme, or it's considered as enzyme-dependent reaction, and these pathways which originate or produce secondary metabolite have relation with the basic metabolic pathways which are similar in animal and plant cells. The basic metabolic pathways are including three type of pathways. One of them is photosynthesis, which is unique for plant cell, and the second pathway is embedding metabolic scheme of glycolysis, which convert carbohydrate and sugars into glucose and produce energy, which is required for plant growth. The third pathway is TCA cycle or Krebs cycle, which is similar in both animal and plant cells. And the importance of TCA cycle is production of energy and production of other intermediates that involved in production of other primary and secondary metabolites. So the reduction of secondary metabolites have relation with uh, basic metabolic pathways, which is a basic for metabolic reaction in all living cells. As we said, production of secondary metabolites have relation with primary metabolism and the three basic metabolic pathways. There are several building blocks originate from these three basic metabolic pathways, considered as precursors for biosynthesis of secondary metabolite. These precursors or building blocks including acetyl coenzyme A or acetyl CoA, shikimic acid, which is responsible for production of aromatic and phenolic compounds such as flavonoid, tannins, coumarins, anthocyanins, and salicylic and so on. The third type of building blocks or precursor is the bionic acid precursors, which is responsible for production of terpene compound, which is similar to the fourth precursors 1-deoxydrigose 5-phosphate, which is known as DOX precursors, deoxydrigose or doxy precursor. These two precursors are responsible for production of terpenoidal compound. And the fifth precursor is amino acid, which is important for production of alkaloids, and alkaloids having containing an aromatic ring or heterocyclic ring beside organic nitrogen, and they have various biological activities, and they have wide distribution in plant kingdom. Each of these building blocks produce several classes of secondary metabolite through unique pathways. In order to understand the biosynthesis of secondary metabolite, we have to remember the difference between animal and plant cells. One of the main differences between these two cells is that plant cells have a rigid cell wall, and the connection between adjacent cell in plant is maintained by primary bits or primary pores that allow transfer or the diffusion of biochemicals, enzymes, and food between animals and between plant cells. Which is actually this cell wall is absent in plant cell. Another important difference between uh, plant cells and animal is that the plant cell contain a chloroplast, which considered as a factory for uh, photosynthesis and production of monosaccharides and small carbohydrate molecules. And another features is that the plant cell contain vacuole, but in a small size when compared to animal cells. Enzymes are playing important role in the biosynthesis reaction for secondary metabolites, and enzymes known as organic catalysts produced by living cells, including plant and animal cells. In their properties, they are colloidal in nature and consist of protein or contain protein as essential part. Also, enzymes characterize that they have high molecular weight and definitely are soluble in ether or dilute solution, but precipitated by highly salt concentration solution and precipitated in organic solvents such as acetone and alcohol. Also, enzymes are inactivated by heat, UV light, and X-ray. From its properties, enzymes are precipitated by action of enzymes, precipitated by action of alcohol. So uh, this explains why we add alcohol to fresh sample immediately after collection to stop the enzyme or role of enzymes, which is may result in hydrolysis of secondary metabolite. The coenzymes are cofactors or chemical groups that aid the role the action of enzymes. One of the important coenzymes that is essential for biosynthesis reaction or play a role in biosynthesis of secondary metabolite is ATP, adenine triphosphate. It is responsible for production of energy. Those contain high phosphate energy bond, and UDP, uridine diphosphate, is responsible for the synthesis of sucrose and glycosides. Nicotinamide adenine dinucleotide, NAD, which is responsible for oxidation and reduction reaction. Coenzyme A, it is responsible for activation of acetate to be active and able to enter the biosynthetic reactions. Flavin mononucleotide also involved in oxidation reduction reaction similar to NAD cofactor. Flavin adenine dinucleotide responsible in transfer of hydrogen atoms in the form of NADH to oxidize cytochrome 60 or to oxidize cytochrome 60. The another important cofactor are thiamine, biotin, and pyroxene, which have a role in decarboxylation reaction. In spite of the importance of enzymes in biosynthesis in the living cells, still the enzymology of secondary metabolic pathways is still uninvestigated or not clearly understandable. But advances in gene technology pave the way for studying in the homology of secondary metabolites. To isolate the enzymes for study, cell culture and plant biotechnology is considered the better source for isolation and for preparation and manufacture of these enzymes. Also, the advancement in protein chemistry and especially chromatography in the process of isolation and studying of enzymes. For one of chromatography techniques that's essential for isolation of enzymes are gel chromatography and affinity chromatography beside dialysis technique. The storage of secondary metabolites, it is one of the important steps in production of secondary metabolite in living cells, because secondary metabolites may have a role in defense for the plant or the plant cells. Also, may have poisonous effect for the plant that may produce lysis of the blood cell. Generally, primary secondary metabolites are biosynthesized in the cytoplasm of the cell or in endoplasmic reticulum, such as protein and alkaloids, or in organelles in mitochondria or some fatty acids. And if these secondary metabolites or these substances are hydrophilic, generally they are exported to vacuum, and vacuum considered as isolated environment from the cytoplasm, and this substance is stored in vacuum until their needs or can be reused or consumed. Generally, water-soluble or hydrophilic secondary metabolites are stored in vacuole or in lacciferous cells, which is considered a type of cells that produce latex, occur in leaf and stem of some plant, or in cell wall, such as tannins, considered as hydrophilic compound stored in cell wall. In lacciferous cells, example of secondary metabolite stored in this type of organelles is some alkaloid and cellogenic glycoside and cardiac glycosides. In vacuoles, many alkaloids can be stored in vacuoles: saponin, glycoside, flavonoid, anthocyanins, and so on. All these are hydrophilic substances stored in vacuoles. For lipophilic substances, they can be stored in cuticle, which is the waxy membrane covering the plant cell. Example of lipophilic secondary metabolite: cuticle waxes, lipophilic flavonoid, and terpenoid. Trichomes also store lipophilic secondary metabolite. One example for secondary metabolite stored in trichomes are monoterpenes and sesquiterpenes beside quinones. And as we know, monoterpene and sesquiterpene constitute essential oil as component of essential oil or volatile oil. Also can be stored in resin duct. Example for this compound, terpenoid and lipophilic flavonoid. Also for lacciferous cells some polyterpenes and diterpenes stored in these anatomical structures. In oil cells, we found some lipophilic anthraquinones and also diacerone compound beside terpenoid. In the plastid membrane, also we find tetraterpenes and ubiquinose compound. This is the general overview for storage of secondary metabolite. If it is hydrophilic or lipophilic, can be stored depending on the nature of this secondary metabolite. Also, the storage of secondary metabolites depends on the type of the plant. If it is annual or perennial plant. For annual plants, which have a short cycle of living or have short age, in annual plants, secondary metabolites are present abundantly in flower, fruit, and seeds. While in perennial species, we find secondary metabolites stored in bulbs or in rhizomes, in roots or in bark of root and stem. Now we will speak about the basic metabolic pathways that essential for production of primary and secondary metabolite, and we will start with photosynthesis. As we know, photosynthesis is a process by which plant, algae, and some type of bacteria, which classified as plant, are converting carbon dioxide into organic compounds. And in plants, photosynthesis occurs in the green organs of plant and mainly within the leaf, and within the leaf the photosynthesis take place in chloroplast. The other part of plant having another role. For example, fruits are responsible for storage of carbohydrates and sugars. Also, the underground system, such as rhizome, responsible for storage, while the roots responsible for absorption of water and minerals, beside transfer of this mineral to the shoot or aerial part. The photosynthesis is divided into two steps. The one step is light reaction, which takes place during the daylight and take place in chloroplast, and the other part of photosynthesis is known as dark reaction or light independent reaction, which take place at night. The second step of photosynthesis is a dark reaction or known as Kelvin-Johnson cycle. In this reaction, in several steps, they produce beta-aldehyde 3-phosphate from fructose and erythrose. From fructose, glucose and sucrose and starch are derived, and from erythrose, aromatic acids are derived. So the Kelvin-Johnson cycle itself consists of three phases. Phase 1, carbon fixation, which involves fixing the carbon dioxide from atmosphere, and then added to the molecule ribose 1,5-biphosphate to produce two molecules of the first compound, اللي هو 3 phosphoglyceric acid or 3 phosphoglycerate, which is in series of reaction known as reduction base produce glyceraldehyde 3 phosphate as the first compound, or the second compound produced in photosynthesis reaction after 3 phosphoglyceric acid. Previously, 3 phosphoglyceric acid and followed by glyceraldehyde 3 steps. In several steps, which is known as phase 3, the regeneration of ribose 5-phosphate that enter the central metabolic pathways to produce fructose and glucose. In fact, the ribose phosphate هو ribose 5-phosphate is 5 carbon monosugar, so monosaccharide. Depending on the first compound used in dark reaction, the plants are classified into 3 grafts. Carbon 3 plant is the first compound produced is 3 phosphoglyceric acid, and carbon 4 plant the first compound oxaloacetic acid and other carbon acids. Carbon 4 plant is a type of plant that grow in semi-arid condition or high light intensity. The class number 3 for this type of plant is known as CAM plant or carasulician acid metabolism, with the first compound produced malic acid, which is also carbon 4 plant consists of 4 carbon atoms and 2 carboxylic group. So this classification is based on that reaction and on the first compound produced. The second pathway of the basic metabolic pathway is the pathway for carbohydrate utilization, also known as embedding Meyerhof scheme of glycolysis, which is metabolize starch and other carbohydrates into glucose. As we know, carbohydrate is stored in plant in the form of starch, while in animals, carbohydrates are stored as glycogen. The pathway for metabolism of glucose is similar in plant and animal. Both of them use starch or glycogen to produce glucose and produce energy. A carbohydrate used for energy production by processes involve conversion to pyruvate, then acetate. And acetate is considered as the first compound that enter TCA cycle or Krebs cycle to produce further precursors and production of energy in cytochrome D450 or in mitochondria. The third pathway in the basic metabolic pathways is TCA cycle or tricarboxylic acid cycle. In this cycle, in one term, one molecule of acetate can be oxidized to uh, two molecules of carbon dioxide and 12 molecule of ATPase. And the first compound that enters the TCA cycle is acetyl or acetate, inactive form acetyl CoA. Acetyl CoA can be converted to uh, citrate, which is carbon 6 compound and consists of three carboxylic groups. Because of this, the name of cycle is tricarboxylic acid cycle. Acetate combined with oxaloacetate compound, dicarboxylic carbon 4 compound to form citrate, or carbon 6 compound with tricarboxylic acid group. Citrate can be dehydrated to cis acetate. Cis acetate converted to also dehydrate to isocitrate, and then the isotrate also is carbon 6 tricarboxylic acid compound. Isotrate oxidized to oxalosuccinate, also carbon 6 oxygenated tricarboxylic acid. Through action of enzyme, oxaloacetate lost one molecule of carbon dioxide to give alpha keto glutarate, which is carbon 5 dicarboxylic acid compound. Oxalo, well, oxyuretate or alpha oxyuretate converted to succinyl CoA by loss of molecule of carbon dioxide and two molecule of hydrogen to convert it to succinyl CoA, which is carbon 4 dicarboxylic acid compound, and then succinate in several steps converted to malate, which is carbon 4 dicarboxylic acid compound, and on loss of two molecules of hydrogen, malate oxidized to oxaloacetate with carbon 4 dicarboxylic acid compound. So the series of reaction occur in TCA cycle producing carbon dioxide and producing molecule of ATPase. Also the intermediate produces such as citrate, succinate, oxaloacetate, fumarate can enter other pathways for biosynthesis of secondary metabolites such as aromatic compound and aromatic alkaloid. The three basic metabolic pathways which we described before, including photosynthesis, embedding Meyerhof scheme of glycolysis, and TCA cycle. All these three basic metabolic pathways are similar in the living cells, and there is a relation between these basic metabolic pathways and production of secondary metabolite. In fact, these three basic schemes are responsible for building up and breakdown of reserve food, and there is a relation between primary and secondary metabolite, and these basic metabolic pathways can be summarized in this figure, starting with photosynthesis, which is responsible for production of 3-phosphoglycerate that enter the other pathways to produce fructose and glucose, which is glucose, can also be considered as part of hydrolysis or glycolysis of glucose of carbohydrate from minimal whole scheme of glycolysis. From glucose as a product of minimal whole scheme glycolysis or as a product of synthesis, a number of secondary metabolites can be biosynthesized, including glycoside, or oligosaccharide, polysaccharide, and so on. Glucose also, through photosynthesis, can be produce glyceraldehyde 3-phosphate, kinetic mean photosynthesis. GPA can produce the pyruvate, which pyruvate can enter the TCA cycle through ethyl-CoA, and itself ethyl-CoA produce a number of fatty acids and polypeptides. Also, ethyl-CoA produce the IBB, isobutyl biphosphate, and dimethyl allyl biphosphate, which is considered as precursors for biosynthesis of many terpenoidal compounds, starting from monoterpene carbon 10 until tetraterpenes or cucurbitaceae. Also, from polypeptide, it's considered as precursor for important secondary metabolite, aromatic secondary metabolite or phenolic, including asteroquinones, naphthoquinones, phenols, and flavonoids. The TCA cycle also, it is source of important intermediate, including alpha ketoglutarate, carbon 4 dicarboxylic acid intermediate. From oxaloglutarate, a number of aromatic alkaloids can be biosynthesized, including purines and tropane alkaloids, which origin from ornithine amino acids and coca alkaloids. Also, pyridine alkaloids can be originated from oxaloglutarate through arginine amino acid as precures. From oxaloacetate as intermediate from Krebs cycle or TCA cycle, amino acid aspartate and lysine are originated, which is considered as precursor for a number of alkaloids, including piperidine alkaloid, leucine alkaloid, and non-protein amino acids. This may be the relation between, in brief, the relation between photosynthesis and one of the basic metabolic pathways and production of secondary metabolites. This flowchart also summarizes the relation between the three basic metabolic pathways and secondary metabolites, also with relation to their organelle through which or in which the pathway takes place. As appeared in the flowchart, the photosynthesis occurs in chloroplast, which produce glyceraldehyde 3-phosphate as ferric compound through fixation of carbon dioxide from atmosphere and release of oxygen as byproduct. 3-phosphoglycerate can produce hexoses such as glucose and other monosaccharides, which is upon glycolysis through enzyme of skim glycolysis can produce glucose, and glucose produce pyruvate and acetate. Pyruvate or acetate can enter the TCA cycle, which takes place in mitochondria to produce energy and this form of ATPase and release of carbon dioxide as byproduct. In mitochondria, through TCA cycle, intermediate a number of fatty acids can be biosynthesized and other polyketated. Also, a number of amino acids can be biosynthesized in mitochondria and complete production of protein in endoplasmic reticulum or their ribosomes. The substance produced from these organelles or through the basic metabolic pathways can be stored in different organelles, in vacuoles, or in cell wall according to their natures. Protein can be stored in vacuole with secondary metabolite. Also, fatty acid can be stored in vacuoles. Some of secondary metabolite can be deposited in cell wall as storage organelles. All this reaction can be governed by enzymes, and the enzymes govern through the nucleus, through genetic material, the DNA in the chromosome, which is can transform the information from the nucleus to the endoplasmic reticulum through messenger RNA strand or messenger RNA chromosomes. Now we'll talk about the origin of secondary metabolite after we know the relation between secondary metabolite and the basic metabolic pathways, also the relation between production of secondary metabolite and organelle, also the relation between the storage of secondary metabolites and the cell organism. For origin of secondary metabolites, almost all secondary metabolites are derived from 5 main building blocks, including acetyl coenzyme A, which is responsible for synthesis of polyketed secondary metabolites, and shikimic acid, which is responsible for vast group of phenolic compounds, including flavonoids, anthocyanins, coumarins, lignans, and so on. The third building block is the hyaluronic acid, which is responsible for biosynthesis of terpenoids or terpene compound. Also, beside the precursor number 5 and number 4, and 1-deoxy xylulose 5-phosphate. So those precursors also responsible for synthesis of terpenes. The last important precursor are amino acids. There are several amino acids responsible for production of alkaloids and aromatic alkaloid and aromatic compounds. The first pathway responsible for biosynthesis of secondary metabolite is acetate pathway, or also known as polyketed or fatty acid pathway. This pathway has acetate as precursor or as building block. It is characterized by that it is occurring in all organisms, The biosynthesis in this pathway start with condensation of one molecule of malonyl CoA with one molecule of acetate CoA to form a simple polypeptide known as acetoacetyl CoA. Again, acetate pathway starts with condensation of acetate precursor with malonyl with the aid of the cofactor coenzyme A to produce acetyl CoA as first precursors produced in acetate pathway. The number of secondary metabolites can be synthesized from these pathways, including antibiotic, tetracycline, and macrolides. Also, some fatty acids can be produced from this pathway, and aromatic compounds such as anthracanic or anthracyclic antitumor agent is biosynthesized through this pathway. Tetracycline is one of the compounds, aromatic compound synthesized through acetate pathway, and tetracycline is discovered as a part of screening program of extract obtained from filamentous bacteria known as actinomyces. In fact, tetracycline have several functional groups, uh, maybe a tertiary amine group, and amide group, besides unsaturated keto groups, and the hydroxy group and keto group considered as gelatin groups. All these functional groups are responsible for antibiotic activity of tetracycline compounds. And the lower oxygenated carbon, including unsaturated keto groups, carbonyl groups في lower part in position 11 وبوزيشن واحد بصير الهيدروكسي جروب في 12 و position 10 indicating that the recursion of this compound is poly beta keto esters or its origin through acetate pathway. The second pathway responsible for production of secondary metabolite is shikimic acid pathway or shikimate pathway. Shikimic acid in nature it is aromatic compound with with dry hydroxy group and one carboxylic acid group. Shikimic acid itself is produced by combination of phosphoenol pyruvate, which is produced through the glycolytic pathway, and one molecule of pentose 4-phosphate, which originates from pentose phosphate pathway. Shikimic acid is precursor for a number of aromatic or phenolic secondary metabolites, including aromatic amino acids such as phenylalanine, tyrosine, tryptophan, which is all considered as precursor for biosynthesis of other classes of secondary metabolite. Also, shikimic acid is responsible for production of simple aromatic acid secondary metabolites such as benzoic acid and gallic acid. Also responsible for production of aromatic aldehyde such as vanillin and benzaldehyde, which are very common in essential oil. Also, shikimic acid is responsible for production of a number of secondary metabolites originate from the amino acid phenyl and alanine. These include phenylbutyrienes, lignans, coumarins, and flavonoids. And the basic feature or the similar feature for this class of secondary metabolite originates from phenylalanine or possess C6 C3 unit, having the chemical structure consists of rhombi side chain with cyclic ring or aromatic hexane ring. One of the characteristic features for shikimic acid is that this pathway is not found in animals, only there in blood. And most of aromatic secondary metabolite pesticide through this pathway are having antioxidant properties. This which characterize plant to be rich with antioxidant compound or phenolic compound compared to animal which devoid from this type of secondary metabolite. This figure shows the relation between shikimic acid pathway and uh, photosynthesis as basic metabolic pathways and the important class of secondary metabolite biosynthesized through shikimic acid pathway. Here shikimate itself is originates from phosphenol pyruvate and erythrose 4-phosphate, which is considered as byproduct or as product of photosynthesis. And gallic acid is one of uh, intermediates produced in the pathway between photosynthesis and shikimate and producing important class of secondary metabolite known as tannins. Shikimic acid itself can be converted into furosemide to a combination of furosemide acid, combination of shikimic acid with acetate ester to produce a number of important phenolic compounds, including naphthoquinones and anthraquinones, and through conversion to anthanylate, shikimic acid can give acridone alkaloids, and through conversion to L-tryptophan, shikimic acid can give indole alkaloid, non-protein amino acids, amine, and oxime. The conversion to this class of alkaloid, tryptophan, is regulated by the enzyme TDC, which is known as tryptophan decarboxylase, and the other enzyme اللي هو streptocidin synthase. This enzyme regulates these classes of alkaloid, including indoles, glucosinolase, and amines, oxines, and NPAS. Also through enzyme BAL اللي هو phenylalanine ammonia lyase. Phenylalanine can be converted to a number of aromatic secondary metabolites, including isoquinone alkaloids, phenyl propanoids, flavonoid, also flavonoid, regulated by the enzyme CHES اللي هو chalcone synthase, which produce flavonoids, and lignan, coumarin, cyanoglycoside, glucosinolate, phenols, and non-protein amino acids. All these originated from crossbase through formation of phenylalanine and tyrosine. From phenylalanine, regulated by the enzyme phenylalanine ammonia lyase, and flavonoid can be synthesized through enzyme chalcone synthase or CHS. One of the important group of secondary metabolites which are biosynthesized through chemical acid pathway, are phenylpropyls. Phenylpropyls are biosynthesized through the intermediate phenyl aniline amino acid, which is hydrolyzed by phenyl-aniline ammonia lyase global to remove amino group, resulting in cinnamic aldehyde, and through reduction to form cinnamaldehyde. From cinnamaldehyde, example of phenylpropane biosynthesized are eugenol, which is the main constituent in clove oil and used as antiseptic. Also, anethol, with the same precursor from cinnamic acid precursor, cinnamic aldehyde. Mycin, also one of phenylpropane biosynthesized from this precursor. It is worth mentioning mycin contain or obtained from nutmeg and from parsley and dill. Another example for phenylpropanes is saffron. Saffron obtained from sassafras oil, which is used for synthesis or partial synthesis of methylene dioxy methamphetamine. It is responsible for the synthesis of abused drug amphetamine. All these phenylpropanes mentioned in these examples are constituent of essential oil and aromatic plant. Not aromatic plant. Another class of secondary metabolite which originate from chemic acid pathway are lignans. Lignans characterized by that they are chemical compound with low molecular weight polymers, where also cinnamyl alcohol is the precursor for lignan. One example of lignans is podophyllitoxin. Podophyllitoxin compound occur in hardwood, leaf, and resinous oxidate of root and bark, and obtained mainly from the genus Podophylla, which belongs to family Podophyllaceae, and podophyllitoxin as lignan has a number of medical applications. It is known anti-cancer agent. Besides, it is considered as preventive and antiviral, anti-dementic agent. Podophyllitoxin, characterized by that, is aromatic compound, obtain aromatic rings with a number of oxygenated functional groups. The third class of phenolic compound to originate from chemic acid pathway are coumarins, and coumarins characterized by that they are all precursor of phenyl-aniline. Coumarins have limited distribution in plant kingdom, occur in families such as family Aviaceae or family Brigley, family Rutaceae, Asteraceae, and family Fabaceae. One of example is Hyllene. Hyllene is isocoumarine or pheromone compound, occur in Amibisnaga family Aviaceae, and it is known as anti-spasmodic or spasmolytic and vasodilator. Flavonoid also a class of important class of secondary metabolites which biosynthesize from chemical pathway, and they are characterized by that all contain C6, C3 unit with C6 group or aromatic ring of originates from chemical acid, and the C3 or bicyclic side chain originates from polyketide pathway. Example of flavonoid is naringine, which is flavonoid glycoside, having bitter and astringent taste that occur in the peel of grapefruit. Another example is quercetin, which is flavanol and present in many food stores and characterized by having strong antioxidant activity similar to a number of flavonoids belonging to this class of secondary metabolite. In the pathway for biosynthesis of flavonoid, the key enzyme that regulate production of uh, flavonoid is chalcone synthase, which control the conversion of tartar unit to chalcone, which is a precursor of flavonoid. Based on the viral nucleus of flavonoid, they are classified into three classes: flavanones, which having the Byron ring with keto group, and another class is flavones, which is unsaturated Byron ring in position 2, 3. Another class is flavanone, which is containing position 3 hydroxyl group. Anthocyanins are responsible for the elegant color of chlorella flower, which is act as attractant for pollinators, also originates from flavanol nucleus of flavonoid. Another compound originated from this pathway is naringine. And naringine belongs to flavanone class. It is glycoside containing disaccharide اللي هما ramos and glucose. والنارنجين dihydrochalcone, which is like the Byron ring, and it's not closed. It is characterized by that it has 1,000 times sweetness of sugar. It's considered as synthetic sweetener. Also, it is glycoside of ramos and glucose. Tannins are water-soluble phenolic compounds characterized by having high molecular weight. Also, it is a class of secondary metabolite originates from chemical acid pathway. They have two classes, hydrolyzed tannins and non-hydrolyzed tannins, which based on their solubility in water and their ability to hydrolysis. Actually, hydrolyzed tannins are glycoside of sugar containing sugar bonds. They are soluble, and non-hydrolyzed tannins are trimers or a polymer produced by condensation of flavonoid. The third pathway for biosynthesis of secondary metabolite is bivalonate and deoxydylose phosphate pathway, and this pathway is responsible for production of terpene compound. In fact, metabolic acid itself is produced from three molecules of acetyl-CoA, and the another precursor, deoxydylose phosphate, arise from combination of two glycolytic pathways اللي هما البيروفيك acid and glyceraldehyde 3 phosphate. And this is the deoxydylose 5 phosphate is carbon 5 compound, while the bionic acid is carbon 6 compound. And the other main difference between methylonic acid and deoxyxylose pathway is that a DOX pathway or DOX route occur in blastid, while methylonic acid pathway occur in cytoplasm. But both of them are responsible for biosynthesis of terpene metabolites, including monoterpenoid, sesquiterpene, terpene, and so on. The main building block for all terpenoid compound is isoprene unit, also known as isobentene, and consists of uh, 5 unit branching hydrocarbon, acetate hydrocarbon, and it is a precursor. Isoprene is precursor for dimethyl allyl pyrophosphate and isobutyl biophosphate, which is considered as a precursor for biosynthetic. And the link between isoprene units to form terpenoid compound is known as isoprene rule, or head to tail joining between two isoprene units to form uh, monoterpenoid compound. For example, this hydro 2 isoprene unit, one considered as tail and another isoprene unit considered as head in position 4 and position 1. The link through 1,4 link produce the first monoterpenoid compound myrosin. The fifth pathway for biosynthesis of secondary metabolite is amino acid pathway, and this pathway is responsible for biosynthesis of alkaloidal compound and some antibiotics, also some non-protein amino acids. Amino acid itself are constructed from intermediate of the glycolytic pathway and from Krebs cycle intermediate. While nitrogen enters to the structure of alkaloid and amino acid through from other source through reductive amination of alpha keto acids. According to the source of that from which amino acids are bioside can be classified into several groups. Firstly, pyruvic acid is considered as non-latrine containing compound from which produced aniline as product of reactive amination to pyruvic acid, and oxaloacetic acid also is intermediate in TCA cycle, responsible for biosynthesis of aspartic acid, and alpha ketoglutaric acid, also intermediate in TCA cycle, produce glutamic acid, and shikimic acid itself, which is precursor for shikimic acid pathway, produce phenylalanine, tyrosine, and tryptophan amino acid. While say phosphoglycerate as a product of photosynthesis or mitoid pathway produce serine and glycine. From this amino acid, a number of antibiotic and alkaloids are derived. A majority of alkaloids are biosynthesized through or from 8 common amino acids as precursors, including ornithine and arginine, lysine, nicotinic acid, phenylalanine, histidine, anthralinic acid, tryptophan, and tyramine acid, or tyrosine. All these 8 amino acids considered as precursor for biosynthesis majority of alkaloids. In fact, another alkaloid may be derived from other precursors, which can be aminated after main ketone has been synthesized, such as purine alkaloid like caffeine, which is originate from purine bases, and aconitine, which is originate from terpenoids, which are product of nucleic acid and those pathway. Also, steroidal alkaloids such as solanidine, which occur in Solanaceae and Liliaceae, which have the basic structure is steroidal nucleus, or steroid here carbon 30 terpenoidal compound. Also, quinine, which is occur in hemlock fruits. It is derived from acetate pathway or acetate derived glycated pathway. After amination, we obtain quinine from acetate precursor. This is why it is overview for biosynthetic pathway for major group of alkaloid, starting from TCA cycle, alpha ketoglutarate or oxalglutarate produce ornithine and arginine, which result in biosynthesis of three classes of alkaloid, pyridine alkaloid, tropane alkaloid, and nicotiana alkaloid. From oxaloacetate through aspartate and lysine, quinolidine alkaloid are produced, beside annabacine as alkaloid belong to this class. From glycolysis, resulting glyceraldehyde 3-phosphate, which is converted to acetoacetyl CoA, which enters the TCA cycle to produce other amino acid, or can produce IBB, اللي هو isobutyl pyrophosphate, that is precursor for steroidal alkaloids. For example, quinine and taxol is class to belong to class of this steroidal alkaloid. Also, the IBB, isobutyl pyrophosphate, can produce another class of alkaloid, including quinoline alkaloid through streptozidine precursor or intermediate. Through chemical acid pathway, also a number of aromatic alkaloids are obtained. Furanoquinoline alkaloid obtained through anthraquinolate and through tryptophan and tryptamine. Aldehyde alkaloids are produced, and through tyrosine from chemical acid pathway, tetrahydroisoquinoline alkaloid is obtained, protoberberine alkaloid is obtained, and benzophenone anthrazine obtained, and avomorphine and morphine as phenylserine alkaloid obtained from tyrosine amino acids. To summarize the biosynthetic pathway and the relation between alkaloid and the precursor, ornithine amino acid is responsible for production of pyridine class of alkaloid, also responsible for production of tropane alkaloids such as atropine and cocaine, Lysine amino acid or acetate responsible for pyridine alkaloids. For example, for this class is choline. Also, ornithine responsible for production of pyridine alkaloids, such as restaurin as example. Lysine amino acid responsible for production of quinolidine alkaloid. An example for quinolidine is luminine alkaloid, and tyrosine responsible for isoquinoline alkaloid production, such as codeine and morphine, and the amino acid tryptophan responsible for production of indole class of alkaloid. And the example for this class is psilocybin, reserpine, and strychnine from Strychnos vomica, and reserpine from Rauwia alkaloid. The last class of secondary metabolites that are biosynthesized is glycosides, and their pathway is unique to some extent because glycoside consists of two parts, consists from sugar and a glycol, and their biosynthesis can be divided into two steps. Step one, formation of a glycol, and the a glycol may be uh, any class of secondary metabolite, maybe terpene, maybe flavonoid, coumarin, alkaloid, lignan, and so on. And the other part is sugar. Uh, Sugars are biosynthesized through pentose pathway or through glycolysis as monosaccharides. So when we talk about biosynthesis of glycoside, we will talk about the origin of a glycol, and through the type of a glycol we determine the pathway through which it is biosynthesized. The next step for biosynthesis of glycoside is formation of glycoside aldehyde bonds through binding of a glycol with sugar moiety. And depending on the type of linkage or what's called aldehyde bond, uh, glycoside can be classified into two classes or many classes actually. For example, C glycoside and the link through CC bond, as in this example, C glycoside, the link between A glycol and sugar through carbon-carbon bond, and O glycoside, which is the link through oxygen carbon bond and known as O glycoside or oxygen glycoside, the link through oxygen bond. An example of important A-glycol part of glycoside is cyanogenic glycoside, cardiac glycoside, anthraquinone and glucosinolates. All these four classes having medicinal application may have effect on the have muscle of having a purgative effect or catalytic action. If we take cyanogenic glycoside as example, we talk about the precursor for biosynthesis of this cyanogenic glycoside. With example, phenylalanine amino acid responsible for biosynthesis of cyanogenic glycoside known as brunacin and tambengenine. These are cyanogenic glycoside produced from phenylalanine. Also, tyrosine responsible for biosynthesis of cyanogenic glycoside known as taurine. Also, it is O glycoside or glucose, and cyclopentanyl glycine is responsible for biosynthesis of didactyl glycoside, and leucine responsible for biosynthesis of pyrrolidine and valine amino acid responsible for biosynthesis of linemarine cyanogenic glycoside. It's worth mentioning all these examples of cyanogenic glycoside are or glycoside of glucose. For cardiac glycosides, they are steroidal, having steroidal nucleus or steroidal aglycone nucleus. And precursor for cardiac glycoside is cholesterol, which is because of biosynthesis of steroidal nucleus. And cholesterol itself originates from the bionic acid pathway, is responsible for biosynthesis of terpenoid and steroid compound. From cholesterol, number of cardiac glycosides are produced. An example is deoxygenin as a glycone responsible for deoxygen cardiac glycoside occur in vegetalis leaf, which is characterized by having cardioactive effect or cardotonic effect, and is used for congestive treatment, congestive heart failure. After we know how the aglycone part of glycoside is biosynthesized by giving example for cyanogenic glycoside and cardiac glycoside. And this second step is the binding of aglycone with sugar moiety, and we talk about O-glycoside as an example. The binding between sugar and aglycone in biosynthetic glycoside is regulated by enzyme reaction, or mainly there are two examples responsible for biosynthetic glycoside: lumma-uridyl transferase and glycoside transferase. In the step for biosynthesis, firstly, UTP, uridine triphosphate, can be linked to sugar 1-phosphate to activate the sugar to enter the biosynthetic reaction. This step is regulated by uridyl transferase enzyme to form UDP sugar, and UDP is a cofactor activating sugar. The second step, the UDP sugar, uridine diphosphate sugar complex can react with aglycone acceptors. ممكن يكون steroidal nucleus أو ممكن يكون cyanogenic nucleus or flavonoid and so on. And the formation of binding between aglycone and sugar is controlled by the enzyme glycoside transferase enzyme, resulting in formation of glycoside with production of UDP as byproduct for this process. At the end of this lecture, we describe the biosynthesis of secondary metabolites, and we know that biosynthesis is a series of biochemical reactions controlled by enzymes and result in production of complex molecule with specialized function, which is known as secondary metabolite, and they having a role in defense for blood, besides their ecological function and medicinal application. Also, we know the difference between plant cell and animal cells, and the importance of organelle of plant cell for biosynthesis of secondary metabolite. Also, we discuss the role of enzyme in biosynthesis, and we describe three basic metabolic pathways involved in production of secondary metabolite, including photosynthesis, including Meyerhoff scheme of glycolysis, and TCA cycle. Also, we know or describe the relation between secondary metabolite and these basic metabolic pathways, because the relation is interconnected between these pathways and primary and secondary metabolite. Lastly, we speak about the origin of secondary metabolite and describe the five pathways responsible for secondary metabolite production, including acetyl pathway, gluconic acid and deoxylase pathway, amino acid pathway. That is all about biosynthesis of secondary metabolite.